What is going on, wonderful people? It's Medicosis Perfectionalis. Welcome back to my microbiology playlist. This video is brought to you by Picmonic. In previous videos, we talked about the gram-positive bacteria versus the gram-negative bacteria. We also talked about fungi, including those that cause superficial mycoses versus those that lead to systemic mycoses. Today, we'll start talking about viruses. This is part 19. You can find the previous videos in a playlist titled Microbiology. The link is in the description. You can find all of these videos in my microbiology playlist as well as in my Picmonic playlist. In part 1, we talked about actinomyces, listeria, corynebacteria, and nocardia. Part 2 was about the Clostridia, including Clostridium tetani, Clostridium botulinum, Clostridium difficile, and Clostridium perfringens. Part 3 was about staph and strep pneumo. Part 4, the rest of the strep and the enterococci. Part 5, Bacillus anthracis, Bacillus cereus, and the mycobacteria. Part 6 was about Neisseria and Moraxella. Part 7, Haemophilus influenzae, Bordetella pertussis, Pasteurella, Brucella, and Francisella. In part 8, we talked about Legionella and H. pylori. In part 9, we talked about Campylobacter jejuni, Vibrio cholera, Escherichia coli, Klebsiella pneumoniae. In part 10, we discussed Pseudomonas aeruginosa, Proteus, and Salmonella. Part 11 was about Shigella and Yersinia. In part 12, we discussed Treponema pallidum, Leptospira, and Borrelia, which cause syphilis, leptospirosis, and Lyme disease respectively. In part 13, we talked about some atypical bacteria such as chlamydia, rickettsia, and mycoplasma. In part 14, we talked about the torch infections. In part 15, we talked about some miscellaneous gram-negatives, at least anatomically, the Anaplasma, the Bartonella, the Coxiella, the Ehrlichia, and the Rickettsia. And starting from part 16, we began to talk about fungi, such as histoplasmosis, blastomycosis, coccidioidomycosis, and paracoccidioidomycosis. These are fungal infections, systemic fungal infections to be specific. Part 17, more fungi, Candida, Aspergillus, and Cryptococcus. In the last video, we talked about Mucorn, Rhizopus, PCP pneumonia, and Sporothrix shanki. Now it's time to talk about viruses, especially DNA viruses. Because as you know, viruses could be DNA viruses or RNA viruses. But I digress. What does microbiology even mean? Ology means the study of. Of what? Of bio, which is life. What kind of life? The microscopic life, the small life. Microbes that can lead to human diseases are divided into bacteria, fungi, viruses, and parasites. But be careful because many parasites are macroscopic rather than microscopic. Ergo, the field of microbiology is divided into bacteriology, fungology or mycology, virology, and parasitology. How do we classify bacteria? Based on the gram stain. We classify bacteria into gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria could be cocci or rods. As for the gram-negatives, also cocci and rods. Purple versus pink. You can learn more about these bacteria by watching my Picmonic videos from video 1 through 15, which you can find in my microbiology playlist. Link is in the comments. If you wish to see more videos like this in the future, drop a petri dish or a bacterial emoji in the comments. I will not forgive myself if I did not tell you that you can tell the difference between bacteria that ferment lactose versus bacteria that do not ferment lactose. The lactose fermenting bacteria appear as pink colonies on McConkey agar, whereas those bacteria that do not ferment lactose appear as white colonies on McConkey agar. Don't ever forget that. Here is a question for the pros. Is E. coli lactose fermenter or lactose non-fermenter? If you know, comment below. Let us learn some basic facts about viruses. First, let me tell you something shocking. Viruses are not actually cells. Because living cells have definitions, they have criteria, they have characteristics, such as living cells should replicate independently. They should be able to make their own proteins on their own. They can produce their own energy, and you can see them by light microscopy. However, viruses cannot replicate independently. Viruses cannot make their own proteins on their own. Viruses cannot produce energy, and viruses cannot be seen by a light microscope. Which means viruses do not check and live up to the 4 fundamental tenets of cell theory. What are the 4 fundamental tenets of cell theory? Number 1, all living things are composed of cells. 2, the cell is the basic function unit of life. 3, cells arise only from pre-existing cells. And 4th, cells carry genetic information in the form of DNA, but viruses are different. Viruses are not really living organisms. You can count them as at the edge of life, or at the edge between life and non-life. Viruses lack cellular structures. Do viruses have a nucleus? No. Do they have cytoplasm? No. Do they have mitochondria? Also no. Viruses are small infectious agents. I said agents. I did not say organisms or microorganisms. First discovered in 1892, they replicate very quickly. That's why everyone is trying to go viral and no one is trying to go bacterial. Viruses cannot make their own proteins independently. Instead, they have to hijack your own nucleus and use your cells as a DNA making machine. For example, the HIV virus is going to hijack your own lymphocytes, which are subtypes of white blood cells. If you want to learn more about lymphocytes, please refer to my hematology playlist. So let's compare between living cells and viruses. Living cells replicate by mitosis or binary fission. Viruses, nope. Living cells have cell membranes. Viruses do not have membranes. Sometimes they have an envelope, sometimes they do not. But an envelope is not the same as a lipid bilayer cell membrane. If we may take the human cell as an example, your genetic material is DNA. That's where your genes are. A gene is a piece of your DNA, and then you can use that DNA to make RNA. We call this process transcription. So you have both. You have DNA and you can use that DNA to make RNA. But for viruses, no, it's either DNA or RNA, not both. That's why we divide viruses into DNA viruses and RNA viruses. In living cells, the proteins are many. In viruses, the proteins are few. Living cells have ribosomes. Viruses do not have ribosome with an exception of the arenavirus. It has ribosomes that do not function or whose function are not known up to this point. The mitochondria are present in eukaryotic cells but absent in prokaryotic cells. In viruses, mitochondria are absent. Living cells have many enzymes. Viruses, the enzymes are few or non-existent. Hey virus, you have the microphone. Tell me about yourself. Well, I either have a DNA or an RNA, not both. I have a protective protein coat. Some viruses are enveloped, others are not. Few proteins, few enzymes, no nucleus, no cytoplasm, no cell membrane. Ribosomes are absent, mitochondria are absent. Viruses cannot produce energy or make their own proteins. Viruses cannot replicate outside your cell or outside of the host cell. They have to be inside your own cell in order to replicate. And that's why technically, from a biological standpoint, viruses are obligate intracellular parasite because they have to hijack you, the host, in order to survive. And they have to live inside your cell. They have to. They're obligate intracellular parasites or agents. We can divide viruses into DNA viruses and RNA viruses. There are bazillion viruses out there. If we focus on the viruses that cause human diseases, we can narrow this down a lot. If you are a student, you need to know about 7 plus 15. 7 DNA virus families and 15 RNA viral families. What are the 7 DNA viral families? Pox, herpes, hepatina, parv, pep, polyadena. This is pox viridae, herpes viridae, hepatinavirus or hepatinaviride. This is parvo, this is papilloma, this is polyoma, and this is adeno. The song is pox, herpes, hepatina, parv, pep, polyadena. I wrote them in this manner intentionally. Why? Because the first row is enveloped. That's why I surrounded by a rectangle like an envelope. But the second row does not have an envelope. These are non-enveloped DNA viruses. The RNA viruses are many, and we shall talk about them in later videos. Today we'll focus on the herpes family of viruses, which is a DNA virus. Today we'll talk about herpes simplex virus, varicella zoster virus, Epstein-Barr virus, and cytomegalovirus, starting with HSV. Let's watch the Picmonic.
Herpes simplex virus. Herpes simplex virus, commonly called HSV, shown by the HARP viruses with signs, is a virus from the Herpes viridae family. This family of viruses is enveloped, shown by the signs made of envelopes. It is a DNA virus, shown by the DNA double helix, with a double-stranded linear genome, the DNA double helix being used as a clothesline. There are two types of herpes simplex virus. Type 1 HSV usually occurs above the waist and can cause oral lesions, typically called cold sores. Other manifestations include herpetic gingivostomatitis, the ginger over the mouth, keratoconjunctivitis, the carrot eyes, and temporal lobe encephalitis, the brain inflames on the temple. After the patient begins to produce antibodies, HSV type 1 becomes latent in the trigeminal ganglia, shown by the 3 gems, and can become reactivated with certain stimuli, including stress and UV light. Type 2 HSV, the HARP sign virus with a tutu, is primarily a sexually transmitted infection that can cause genital vesicles, portrayed by the genital gentleman wearing a vesicle vest. Infants that pass through the vaginal canal in a female with an outbreak of genital herpes can lead to neonatal herpes, shown by the baby, which is a torches infection. Type 2 HSV becomes latent in sacral ganglia, the sac in this picture. HSV infection can be diagnosed via visualization of organisms on tank smear, the Z-tank, which is a scraping of an ulcer base for evidence of multinucleated giant cells, the multinuclear shells. Cowdrey body A inclusions, here the drying cow, are also present in herpes infection. Acyclovir, the apple cyclops, is an antiviral medication commonly used as treatment.
Herpes can lead to oral sores, genital sores, encephalitis, gingivostomatitis, and more. Next is varicella zoster virus, which can lead to chickenpox in young people and shingles in old people.
Varicella zoster virus, the varsity Zorro virus, is the third of the 8 viruses in the Herpes viridae family, shown by the HARP virus on the 3 tree. This virus is an enveloped DNA virus, shown by the DNA double helix, with double-stranded linear genome, the double helix used as a clothesline. Primary infection results in chickenpox, shown by the chicken in the box with a rash. Adults that get infected with primary infection commonly have more severe disease manifestations and are more likely to experience complications such as pneumonia, the nude mona, or encephalitis, the brain on fire. Even when clinical symptoms resolve, the virus remains dormant in the trigeminal and dorsal root ganglia, shown by the 3 gems on the door, and can reactivate later in life to produce shingles, represented by the roof shingles. The organism can be identified via sank smear, the Z-tank, with visualization of multinucleated giant cells, the multinuclear giant shell, or the presence of Cowdrey A inclusions, here the drying cow. Antiviral drugs like acyclovir, the apple cyclops, can be used to reduce the severity and duration of infection. So let's recap. Varicella zoster virus is the third of the 8 viruses in the Herpes viridae family. It is an enveloped DNA virus with a double-stranded linear genome. Primary infection results in chickenpox, and adults that get infected with primary infection commonly have more severe disease manifestations and are more likely to experience complications such as pneumonia or encephalitis. Even when clinical symptoms resolve, varicella zoster virus remains dormant in the trigeminal and dorsal root ganglia and can reactivate later in life to produce shingles. Varicella zoster can be identified via sank smear, with visualization of multinucleated giant cells or the presence of Cowdrey A inclusions. Antiviral drugs like acyclovir can be used to reduce the severity and duration of infection.
Next is Epstein-Barr virus, which causes infectious mononucleosis, otherwise known as the kissing disease, because it's transmitted through saliva.
Epstein-Barr virus is portrayed by Einstein-Barr virus, as he's trying to do a pull-up on the bar. This virus is also called human herpes virus 4, the HARP with the 4 fork. The virus is double-stranded, the DNA double helix, and linear, portrayed by the line. This virus is enveloped, represented by the envelope in the image. Remember that this virus can cause infectious mononucleosis, illustrated by the monkey nuclear, for mononucleosis. Epstein-Barr attaches to CD21 receptors on B cells, illustrated by the CD with the 21 blackjack cards on the basketball. Epstein-Barr virus can also lead to the development of nasopharyngeal carcinoma, the nose pharaoh with the carcinome, as well as Burkitt's lymphoma, the bucket with lime foam. The development of Hodgkin's lymphoma, shown by the hog king with lymph-lime foam, has also been associated with Epstein-Barr. So in summary, Epstein-Barr virus is also known as human herpes virus 4 and is a double-stranded DNA virus, which is linear and enveloped. It is the pathogen which causes infectious mononucleosis. It works by attaching to CD21 in B cells and can lead to nasopharyngeal carcinoma, Burkitt's lymphoma, and Hodgkin's lymphoma.
Let's talk about infectious mononucleosis in detail. By the way, in most cases, infectious mononucleosis, or simply mono, is caused by EBV. But in a minority of cases, infectious mononucleosis is caused by CMV. When the infectious mononucleosis is caused by EBV, it will give us a positive mono spot test. But when the infectious mononucleosis is caused by CMV, we get a negative mono spot test.
Remember infectious mononucleosis as the story of the monkey nuclear plotting world domination and releasing his chemicals into the water to attack unsuspecting citizens. This contagious disease is typically spread via oral secretions, giving it the name the kissing disease, and is commonly caused by the Epstein-Barr virus, making it a herpes-like viral infection, shown by the herpes harp virus attacking citizens. His bite causes the sleepy guy to fall into a deep slumber, as generalized fatigue can persist for 2 to 4 weeks in patients with mono. A low-grade fever may present, shown by the fever beaver spitting flames, as a sore throat is another common symptom. This disorder may be mistaken and treated as a strep-type upper respiratory infection due to the similarity in these symptoms. The beaver is smacking a bystander, causing head and body egg lumps, as patients may complain of head and body aches persisting for several weeks. Upon assessment, patients classically present with an enlarged liver and spleen, known as hepatomegaly and splenomegaly, depicted as the liver and spleen balloon. Due to the possibility of splenic rupture, patients must be advised to avoid all contact sports. Swollen lymph nodes in the neck and armpits is also common. The swollen limes in the man's neck and armpits. He throws his limes into the monkey's nuclear smokestack, causing a reaction that produces a rash, as a full-body rash often develops if patients are mistakenly treated for an upper respiratory infection with antibiotics. Considerations for patients with mono include the development of a suppressed immune response, shown by the suppressed immune responding with a positive monkey spot test, as diagnosis of this virus is with a positive mono spot test, which detects heterophilic antibodies in the patient's serum. So to review, infectious mononucleosis is caused by a herpes-like viral infection. Symptoms include fatigue, fever, sore throat, head and body aches, enlarged liver and spleen, swollen lymph nodes in the neck and armpits, and possible rash. Patients may have a suppressed immune response for some time, and a positive monospot test confirms diagnosis.
Epstein-Barr virus, or EBV, shown as Einstein doing a pull-up on a bar, and monkey nuclear virus, is a virus that can cause a range of clinical illnesses. A common manifestation is the disease mononucleosis, shown here as the monkey with nuclear sign. Clinical signs include fever, illustrated as the fever beaver, hepatomegaly, the liver balloon, splenomegaly, the spleen balloon, and pharyngitis, the pharaoh. Patients also often have posterior cervical lymphadenopathy, illustrated by the post-terrier with cervical spine showing with the lymph-lime add pluses. On blood smear, atypical lymphocytes will be seen, shown by the ATP with an atypical Lyme lymphocyte, which are larger than normal lymphocytes. Heterophile antibodies will also be detected, depicted by the hat of phials. The differential diagnosis includes streptococcal pharyngitis. So, when mononucleosis patients are accidentally treated with ampicillin, they will develop a rash, represented by the rash on the fever beaver being examined by the dermatologist standing on the amp pencil. Because EBV infects lymphocytes and can alter their DNA, patients rarely can develop Hodgkin's lymphoma, shown by the hog king with lymph-lime foam. So, in summary, Epstein-Barr virus is the cause of mononucleosis. Clinical signs include fever, hepatomegaly, splenomegaly, pharyngitis, and posterior cervical lymphadenopathy. On labs, atypical lymphocytes and heterophile antibodies will be seen. When mononucleosis is accidentally treated with ampicillin, patients develop a rash. Lastly, EBV infection can rarely result in Hodgkin's lymphoma.
And finally, cytomegalovirus.
Cytomegalovirus is depicted in this Picmonic as the cytomegavirus. Commonly called CMV, cytomegalovirus is an enveloped, shown literally as the envelope, double-stranded DNA virus, the DNA double helix. It is also called human herpes virus 5, illustrated by the HARP virus with the 5 hand. When diagnosing cytomegalovirus, remember that unlike Epstein-Barr virus mononucleosis, cytomegalovirus mononucleosis shows non-heterophile antibodies, the non-hadophiles, and thus, patients have a negative monospot test. On microscopy, visualization of owl's eye inclusions in a cell, the owl with giant eyes, is highly specific for cytomegalovirus infection and can aid in disease diagnosis. This virus can cause a variety of infections in humans, including congenital torch infections in infants, shown by the torch. In adults, infection can cause mononucleosis-like symptoms, the monkey nuclear. In the immunocompromised, cytomegalovirus can cause pneumonia, portrayed as pneumona. Don't forget that CMV is also a cause of AIDS retinitis and esophagitis, shown as the band AIDS on the red tin eyes and sarcophagus. Finally, because this virus has many opportunistic features, we often see CMV infections in transplant recipients due to their chronic immunocompromised state, and you can recall this with the trained plant. So let's review cytomegalovirus, or CMV. This is an enveloped, double-stranded DNA virus, which is also known as human herpes virus 5. Remember that unlike Epstein-Barr virus mononucleosis, labs show non-heterophile antibodies, and thus a negative monospot test. On microscopy, we see owl's eye inclusions in cells. Signs and symptoms of cytomegalovirus infection include congenital torch infections, mononucleosis, and in the immunocompromised, pneumonia and AIDS retinitis and esophagitis. Additionally, CMV infection is seen more often in transplant recipients.
If you want some colorful pictures to help you categorize and classify the bacteria and the fungi, Picmonic has many resources. After you watch these Picmonics, you can actually quiz yourself. There are several multiple choice questions after each Picmonic. Multiply that by 1,800 Picmonics and you easily get more than 5,000 multiple choice questions on this website. By the way, can you tell me the answer to these questions? Comment below if you know. You can browse these Picmonics by your favorite book, by subject, and by system. Every single day they give you a quiz based on the topics that you have reviewed before, so that you can refresh your memory. This is my methodology of understanding and mastering these Picmonics. First, I watch the animation, then the story animation, and then I watch the animation again. I pause and stare at the picture. I close my eyes and use my imagination to put each item and each character in its place. Then I open my eyes and see how I did. I solve the quiz. I get a piece of paper and write down the place of the characters. I revisit the same Picmonic on day 2, on day 50, and on day 30. This use of spaced repetition helps me remember these Picmonics for a very long time, sometimes years or decades. For example, I still vividly remember the Picmonic on tuberous sclerosis, which I've watched more than 10 years ago. You can try Picmonic for free. You can use it on your desktop computer or on a mobile app or even on a tablet. They use visual, audio, storytelling. You can read a script. You can practice spaced repetition. There are multiple choice questions after each Picmonic. You can even create your own Picmonic and watch new Picmonics made by other students just like you. As we do towards the end of each video, we review and compare between these viruses. So here we have HSV, VZV, EBV versus CMV. Let's go. The characteristics. All of these are DNA viruses rather than RNA viruses. All of them are double-stranded and not single-stranded. All of these viruses have an icosahedral capsid rather than a helical capsid. And in all of them, the DNA structure is linear rather than circular. Next, let's talk about diseases. HSV can lead to keratoconjunctivitis, gingivostomatitis, herpes labialis or the cold sore, herpes genitalis or the genital sore. Herpes simplex virus can also lead to temporal lobe encephalitis. Remember that we use the zinc smear in order to diagnose herpes, and of course you can use PCR. Here's a mnemonic to help you remember that HSV causes temporal lobe encephalitis. I want you to get your hand and put it on the side of your skull. Put it at your temple or temporal region and say with me, thank heavens I do not have herpes. Next, varicella zoster virus tends to cause chicken pox, usually in young people, and shingles, usually in old people. One of the complications of shingles is post-herpetic neuralgia. Let's break that down. Algia means pain in the nerves that happens after herpes, post-herpetic neuralgia. Algia means pain, but analgesic is anti-pain or the absence of pain. Shingles usually follows one dermatome, not two, on one side of the body, not both. There is usually pain followed by the rash, and the rash is unidermatomal or monodermatomal ipsilaterally on one side only. And then after the rash, I can develop post-herpetic neuralgia. So it's pain, then the rash, then pain. What should a good doctor do? A good doctor should recognize the pain before there is a rash. Oh, doctor, it hurts like here, and it hurts, and it follows one dermatome, and it's on one side of the body, not both, and it's on the trunk. What should I do? Before it becomes a rash, you should start the treatment to prevent it from progressing. That's a good doctor right there. A worthless doctor will just give aspirin or ibuprofen. Oh, take that for the pain and go home. Don't you have any self-respect? When was the last time you opened a book? Next, Epstein-Barr virus. It can lead to monospot positive infectious mononucleosis. EBV can lead to all kinds of lymphoma, including Hodgkin's lymphoma and non-Hodgkin's lymphoma. And the non-Hodgkin's lymphoma includes diffuse large B-cell lymphoma, which includes primary CNS lymphoma. EBV can also lead to oral hairy leukoplakia as well as nasopharyngeal carcinoma. Next, cytomegalovirus. What does it do? It causes monospot negative infectious mononucleosis. It can lead to retinitis, especially in immunocompromised persons. It can lead to CMV esophagitis and colitis, inflammation of the retina, inflammation of the esophagus, and inflammation of the colon or the large intestines. Treatment for all of them is acyclovir or valcyclovir or famciclovir, but for CMV it's different. It's not acyclovir, but rather ganciclovir. What if this CMV is resistant to ganciclovir? Then you should use foscarnet instead. For the infectious mononucleosis, the treatment is supportive care, and the person should avoid contact sports because of the splenomegaly that can rupture. As for the cancers, you will need chemotherapy, surgery, radiation, and or a combination of all of them, depending on the cancer, its grading and its staging, etc. To learn more about cancers, check out my oncology playlist and my pathology playlist. Let's review. Herpes simplex virus is an envelope virus. It is double-stranded DNA virus. HSV1 is above the waist, usually. HSV2 is below the waist, usually. I said usually, not always. HSV1 causes gingivostomatitis, keratoconjunctivitis, and encephalitis of the temporal lobe. The virus remains dormant in the trigeminal ganglion. HSV2 causes genital vesicles. It causes vertical transmission or neonatal herpes from mother to baby during labor and delivery. And it's one of the torch infections, which we have covered in a separate video in this microbiology playlist. HSV2 remains dormant in the sacral ganglion rather than the trigeminal ganglion. How do we diagnose it? Zinc smear or PCR. Look for multinucleated giant cells. You can find Cowdery A inclusions and you treat with acyclovir. Next is VZV, which is also known as HHV number 3. Is an enveloped virus that is double-stranded. Diseases include chickenpox, pneumonia, encephalitis, and shingles after it remains dormant in the trigeminal and dorsal root ganglia. Diagnosed with the zinc smear, look for multinucleated giant cells and Cowdery A inclusions, and you treat with acyclovir again. Next is EBV virus or Epstein-Barr virus, also known as human herpes virus number 4, is also double-stranded DNA and linear. Does it have an envelope? Yes, it does. What does it cause? Infectious mononucleosis, where the virus attaches to CD21 in B lymphocytes. That's the receptor for the virus. Other diseases include nasopharyngeal carcinoma, Burkitt's lymphoma, and Hodgkin's lymphoma. Next, infectious mononucleosis. It has fever. It has hepatomegaly and splenomegaly. It has pharyngitis. It has posterior cervical lymphadenopathy. We find atypical lymphocytes on peripheral smear. In the lab, it gives us heterophile antibodies. This is what you call a positive monospot test. The rash develops if the patient receives ampicillin, so do not give ampicillin. Mononucleosis is typically self-limiting. However, it increases your risk of Hodgkin's lymphoma later in life. Finally, we have the cytomegalovirus or CMV. It's an enveloped virus. It's a double-stranded DNA virus, known as human herpes virus 5. Expect non-heterophile antibodies, so it's a negative monospot test. You can find the owl eye inclusions on biopsy. CMV can lead to a congenital torch infection. It can cause infectious mononucleosis, it can cause pneumonia, and it can cause esophagitis and retinitis in immunocompromised persons, such as AIDS patients and organ transplant recipients because they receive immunosuppressive therapy. Use the link in the description box to try Picmonic for free. And if you decide to subscribe, you get access to more than 1,800 different Picmonics, including Picmonics for all of the high-yield bacteria and fungi and viruses and parasites. And that's just microbiology. You can also find Picmonics for anatomy, physiology, biochemistry, pathology, psychology, internal medicine, and even statistics. My favorite Picmonic subjects are microbiology, pharmacology, genetics, and obstetrics and gynecology. Whether you're studying to be a doctor, a nurse, physician assistant, nurse practitioner, pharmacist, PT, OT, a dentist, or a paramedic, Picmonic has a solution for you. You can download the app for free right now on your phone or tablet and go to picmonic.com/VIP hookup/medicosis and they will hook you up. Thank you guys for watching. Thank you Picmonic for sponsoring this video. Click on this link in the description box. We'll see you next time. As always, be safe, stay happy, study hard. This is Medicosis Perfectionandus, where medicine makes perfect sense.